Becky.
Can you comment on the length of time frequently involved in these studies and any efforts to shorten
that? Right. Most of the studies are a year, 12 months. However, some of the ones that we're designing now are as short as 6 months. And we think that, and this is just in the last basically year or 18 months that we've come to the realization, basically with the statisticians' help, that you can actually get all the information you need to see if, with the ALSFRS, if the drugs have an effect on the deterioration of the slope of that ALSFRS in the first 6 months. The second 6 months, 7 to 12, didn't really add anything. So in the Rasagaline trial that we're designing, that's going to have a placebo. But to do that, you have to have a placebo arm. But the Rasagaline study that we're designing that has a placebo arm, I believe it's 6 months. That's where we're doing our primary efficacy. And then we have another 6-month compassionate open label use. So I think that the ALS research community, with the help of our statisticians, looking at all the data, and we have a lot of data now on negative trials, and we basically have data on 800 patients that have been entered in trials over the last few years that were in the placebo group. So we know how fast that slope deteriorates. And I think because of all the contributions of patients that have been in these trials for the last 5-6 years, with that database, the statisticians now are telling us that in most of the studies, we can model it for a 6-month trial. And the Neurology study is 6 months. Right, Maureen? Yeah. Well, and the treatment part. No, and I don't remember the CNOP study. Did they keep theirs 12 months? Some of the protocols that were written a few years ago are still 12 months, but we're trying to do it 6 months. I don't know if we can do it any quicker than that, unless we get a home run.
No, that cut it in half.
Right. Yes, Sally?
Could you speak to the research consortiums that...
Yeah. So the only way to do these sorts of trials where we have 400 or 500 patients in a trial, or even if you're just shooting for 100 patients, I mean, even though we see a lot of patients here, each center can only enroll a max about 15 to 20 patients in a trial. And you need to get an answer several hundred patients. And so you have to have a consortium of ALS research clinics around the country. So we've been involved in working with these consortiums from coast to coast. So there's a West Coast one called the Western ALS Study Group, or WALS. And then that's led by Dr. Miller, and I'm the co-director of that in San Francisco. He's in San Francisco. And then there's an East Coast group, Northeast ALS Study Group, NEALS. And we're members of NEALS too. And because we're right in the middle in Kansas City, we can be a member of both groups. But actually, in reality, the group membership now is sort of opened up, and you can be in New York and be a member of WALS and vice versa. But there are groups of us that get together to think of these protocols, think of protocol design, and then we work together in a very collaborative way. And then if a drug company has a drug that comes down the road that they want to try, they know to come to our groups, and then we can put these drugs into trials into place. Yes?
If you're involved with a study, after the study's over, is there any way to find out whether
yeah. Yeah, no, that's part of the deal when a patient signs a consent, is that at some point we will tell them if they were on active or placebo. And so that's a promise that we make, and we always can keep that promise. The problem is that, and we have to be very honest with patients up front about this, that we can't break the code and release the results of which one you're on until the entire study is over. They won't, the statisticians and the people that design the studies, you can't look after you finish your 6 months to see what you were on. All 300, 400 patients have to finish, and then we can tell them. So it's a bit frustrating for patients and us, and it's often 6 to 12 months after the trial, after you have finished your involvement in the trial. Now, sometimes what the drug companies will do, and even in the trials that we think up on our own, we call those investigator-initiated trials, you'll be in the placebo versus active for 6 months or 12 months, and then there will be a compassionate use extension where you're on the active drug indefinitely until the answer is found. Did it work? And we have one trial like that now, the Canap Primipaxil trial, where patients have stayed on it while we're waiting for that final results. And sometimes during the course of that phase, when you're on open label, they'll give us the key to the placebo control. But it takes a bit of time, and we have to tell patients and families that up front. Fast studies like the Botox study we did quickly, and so we were able to tell them in a few months after the study was over. But these long ALS trials, it takes a long time. Maureen Walsh back here is our unbelievable research coordinator, one of the best in the country, and she almost single-handedly, with Laura's help and with the help of some of the other younger folks that have joined our research team, gets patients in these trials and nurtures them through it and sees them in their study visits. And I've never seen one individual handle so many ALS research patients in trials. Pretty amazing. Okay, I'll stop there. If you have more questions, we'll address them at the end. But Dr. Swerdlow, why don't you come on up? Dr. Swerdlow is professor of neurology, and he came to us from the University of Virginia 4 years ago to start a laboratory program in the Department of Neurology to study why patients develop neurodegenerative diseases. And he works in Alzheimer's, Parkinson's, and ALS. So he's going to tell you a little bit about his thoughts on why folks develop these diseases.
Thanks, Rick, and thanks for being here. It's a pleasure to be here and to talk a little bit about what I do. I'm a physician scientist, and my career goal really is to understand and work on treatments for neurodegenerative diseases, ALS obviously being a neurodegenerative disease. There are commonalities between the different neurodegenerative diseases, ALS and Alzheimer's disease and Parkinson's disease. And one of these main commonalities comes down to a part of the cell called mitochondria. And this is a picture of a mitochondria. It's like a little spaceship here shown floating around in a cell. And mitochondria are important in terms of generating energy within the cell. And they also help a cell decide whether it's going to survive or whether it's not. So we think it's very important in neurodegeneration, and certainly people have observed that there are changes in the mitochondria of people who have ALS. We kind of got on this bandwagon very early in the mid-1990s. We were one of the first groups to identify mitochondrial problems in people with ALS, and I want to talk to you about an experiment that we did and that we still do here at KU. We have cells that we grow in the lab. They're like brain cells, they're like neurons, and we've treated them to remove their mitochondria, and then we can take a blood sample from a patient and we move their mitochondria into these cells. And so now we have a cell that has the mitochondria from the person that we want to study their mitochondria. And this is a picture of kind of what it really looks like. So these are neuron cells growing in a dish, and they have mitochondria from a person with ALS. And this is you can stain it with different things and different dyes and really take really fancy pictures. And here the red dots are all mitochondria from a person with ALS. And what we found when we study these cells, because it's kind of like it's one of the best ways to model the disease, at least I think so. What we find is that the mitochondria look different when they've come from a patient with ALS. And we find that these mitochondria, when they come from someone with ALS, that they have an altered ability to make energy. And we also find that the mitochondria are overproducing something called free radicals, something I think most people are familiar with, that they've heard about, oxidative stress. And the mitochondria have a reduced ability to do other functions that they normally perform, such as store charged molecules within cells. And they also, we've recently, in a recent study we've shown, places cells in a pre-degeneration state. So obviously we'd like to think about ways to try and reverse these changes that we can see. And Dr. Barohn mentioned that Dr. Wong had come up with this idea to study this drug called Rasagaline in ALS patients. And one of the things that makes Rasagaline interesting to people in the neurodegeneration field is that at least the people who produce this drug have reported data that in which they claim that the Rasagaline may actually help fix problems in mitochondria. And they are wondering whether it might be what we call a neuroprotective drug, and if it is, could it be neuroprotective because it works by fixing mitochondria? And the trial that Dr. Barohn just mentioned that is getting underway here with Rasagaline, one of the things that we're going to do is we'll be looking at blood samples from people who are in this trial, and we'll be able to look and see whether the Rasagaline is actually having an effect on their mitochondria. So it's what we would call a biomarker extension of the study, and it's basically trying to look to see is the Rasagaline, is the drug actually hitting the target that it's supposed to be hitting or that it's claimed to work through. So one of the things that my lab is very actively engaged in, and has been for some time, is figuring out ways to fix mitochondria. And obviously, we're trying to develop drugs that will do this. And in our efforts, we are informed by certain interventions that you can do in a laboratory or outside a laboratory, such as caloric restriction, which means eating less, a lot less, and exercise, because we know that when people eat a lot less or when they exercise, that this seems to have beneficial effects on their mitochondria. But the problem is that when you calorically restrict, the part of the body where the mitochondria really get the benefit is in the liver. And when you exercise, the part of the body where the mitochondria really benefit is the muscle. But we feel if we can develop an understanding about why these effects are happening, that will help us develop drugs that will be able to go to the brain and actually induce this in the brain. And so I thought I'd just show you some short videos of how we go about trying to do this. This is an example of a mouse on a treadmill. And so I have a graduate student who's getting her PhD on exercising these mice, and then we study them, and we try to figure out what is it about the exercise that improves their muscle mitochondria, and does it affect mitochondria elsewhere in their body as well. Here's just another example of this. A mouse on a treadmill. We have a little mouse gym, and the mice run for 45 minutes a day.
It's right down the hall, too, right?
It's right. It's right in this building, and here's Leslie, the graduate student. She's got a little brush, and sometimes the mice get a little lazy, and just by tickling their tails, it just eventually. And we have like I said, we really do have a whole gym. So this is a whole set of treadmills. Series of treadmills, and we could, so the mice are all working out, they have, I guess, CNN on, and if the mice decide that they don't want to run, there's a little, at the bottom here, there's a little mesh, and they actually get a little shock, and you can see them jump back onto the treadmill, and it's really not a bad shock at all. Some of them prefer to actually sit there and be shocked and run, but we really learned a lot about how what mechanisms are actually improving the mitochondrial function in these animals, and we've gotten some insights, and we've actually made some progress in terms of developing ways to be able to accomplish this in the brain, have the brain actually make more mitochondria. So with that, I'll just say thank you.